Yeah, in the previous sections we have seen how the protoplast can be isolated from different plant parts, and we have isolated them, we have purified them, we have measured whether they are viable or not. Next is the culture. Before culturing, we have to make sure that in our culture media suitable density of protoplast fits in. That's why we use the hemocytometer, onto which we load the solution or the media which contain number of protoplast. We literally count them, and once we have made sure, okay, this is the density at which the protoplast is going to survive on the media, that density after adjusting, we go for the culture. Then before the culture, even we need to separate the protoplast, and the during the isolation itself, the cell has to be washed. Then what are the constituents to wash the cell as well as protoplast? The media is known as cell protoplast washing or CPW solution, which consists of different salts, K2PO4, KNO3, then calcium, magnesium, then potassium iodide, copper sulfate with the pH 5.8, which is the normal pH used for the culture. Then you can notice the CPW 13M and 21S. What do you mean by 13M? 13M is nothing but 13% mannitol or 21% sucrose. These two different solutions are normally used for the cell protoplast wash solution. Protoplast isolation and handling should be done using this solution, as it stabilizes the osmoticum of the protoplast and maintains the cell without bursting. For centrifugation, normally the CPW 13M is used, and next the CPW 21 is used, which makes the protoplast to form a band by floating, and debris and intact gels are settled at the bottom. This is the actual protoplast band, and remaining thing would be separated. Maybe some of the portion which the cell debris and all, they may get settled at the bottom of the centrifuge tube, whereas the viable protoplast, which forms an interfering band between the two solutions, and we can easily isolate these protoplast band even using the spatula or some other aids. Then after that, the protoplast has to be cultured. We have to dilute and adjust the protoplast using the hemocytometer. After that, we can employ the culture method, which could be solid culture. The common media formulated by Nugget and Takabe, the scientists, which contain 1% sucrose and 13% mannitol at the 2x agar concentration. The media is very solid compared to the normal media what we are going to use in the laboratory. After the media has been cooled around 37 degree, after the agar has been dispensed, the temperature has to be brought down to 37 degrees, which is the normal body temperature of human beings. That supports the viability of the protoplast as well. We have to mix the protoplast into the media before dispensing, and after that we can adapt into the Petri plates and invert the plate after solidification of culture. Now light intensity should also be very less compared to the 5000 to 10,000 lux. It is very low, 500 lux, which is sufficient for the isolated protoplast. Then during subculture, the mannitol can be gradually brought to the 0%. By the time the cell, as it forms the cell wall, it could protect from the osmotic pressures and all, so mannitol can be reduced, and it is supplemented with the sucrose for the normal levels. Then this is the actual media which was proposed for the tobacco mesophyll protoplast when they have isolated. You just notice different salt solution which contain the NPK and other major and minor salts. Then just notice the presence of sucrose, which plays an important role. When the protoplast have been isolated and they have acquainted to the media, gradually the mannitol has been completely removed and the normal sucrose level has been used. Gradually notice even the mannitol will be reduced. Now different methods involve. Droplet method, we can suspend the protoplast in media and place them on the Petri plate as a droplet, and we can culture them in a normal tissue culture laboratories. And the other method involves the co-culture and conditional medium. Already we may have cultured the protoplast previously. To the same cultured protoplast, we can mix up the freshly isolated protoplast and we can place them on the same media, solid media. What is the advantage? The pre-cultured cells or protoplast, they provide fresh protoplast growth factors for their normal growth and survival, because they might have already secreted and made sure that the media supports the new growth. which is very difficult to establish at the inception. So pre-cultured or conditioned media is with the advantage or an edge. Then the other method involves feeder layer or nurse culture, wherein the fast growing cells or the protoplasts which are arrested mitotically using a chemical or the physical agents like the gamma radiations, they are literally lit on the protoplast so that to arrest them or to kill them. Metabolically they are not killed, only their cell division will be halted. Onto those inactivated cells or half killed, you can say half killed cells, we can place the freshly isolated protoplast, and before placing we can even make sure that a thin tissue paper can be kept before placing the newly isolated protoplast. Another method involves hanging droplet method, wherein a droplet of protoplast solution placed on the Petri plate, and after placing we can literally invert the Petri plate. Bottom plate is placed with the sterile distilled water to maintain the humidity. Humidity also plays an important role for the growth and development of isolated cells in the plant tissue culture. So distilled water placed at the bottom plate maintains such high level of humidity, and we have to make sure that the entire thing Petri plate has to be sealed. Another type is nothing but bead culture or entrapment, wherein different inert materials like the alginate, carrageenan, agar-agar, etc. could be used to make beads in which protoplasts are entrapped or trapped. Then these beads can be literally suspended in a liquid media, kept on the shaker for constant agitation and supply of nutrients. Then the advantage is that less heat stress compared to the solid culture, as the liquid protects from such a different stress. Then after culture, how to make sure that the protoplast is responding? The first sign of the cell responding or protoplast cell is responding is that the wall has to form. Before any response, if there is no wall formation, then the cell viability, though it has been there initially, if the wall formation will not happen, then ultimately the protoplast will die. So the first sign of life is within 1 week the cell wall has to be completely formed. If it fails to do so, then the protoplast viability will be lost gradually. Then gradually the spherical loss, when you have isolated a protoplast, they literally look like a globular mass or a spherical shape. Now compared to the plant cells which are angular, if the protoplast they fail to become angular. What do you mean by angularity? When the protoplast secretes the cell wall, they actually form the different angles, and this indicates, okay, I have synthesized the cell wall and I am normal. This is nothing but the losing of the spherical shape. If they retain their globular shape, which is the characteristic of any liquid for that matter, that indicates I am not responding. So spherical shape, if it is lost, it is a visual indicator that the cell is responding. Then only the cells which have formed the wall, they are able to divide. Then cell division and callus formation could be the next phase, wherein the cell, if it is responding to the media, rapidly divides, redivides and forms the callus. Plating efficiency would be extremely low and in some of the cases, but in some cases extremely high depending on the species. Plating efficiency is nothing but number of dividing colonies per field divided by number of live protoplast at plating. After 2 weeks, maybe after a fortnight, multicellular colonies will form, which is nothing but a microcallus. After a month or so, macroscopic colonies can be transferred, which have been formed onto the media to the new solid medium. Then the plant may regenerate slowly. Mini callus colonies which are grown on callus induction media. This callus is transferred to the regeneration media with the appropriate oxygen as well as cytokinin concentrations, which again depends on the species, will result into an organogenesis wherein shoot and root will form or result in somatic embryo formation. Let me continue in the next class regarding the general properties of the protoplast and all.